When we talk about protein structure, it's important to understand these four various structures that are possible amongst polypeptides or proteins. So the primary protein structure is very simple. It's essentially just the sequence of amino acids. So it could be alanine, tyrosine, serine, arginine, etc. So it's just the sequence of amino acids. So the secondary protein structure, it has two possible forms. It can either be an alpha helix or it could be a beta plated sheet. In both of these forms, the main force that provides us with these two structures is the hydrogen bonding of the oxygen and nitrogen within the peptide bond. So if we can remember, the oxygen has a partial negative on the peptide bond and the nitrogen has a partial positive. I can draw it out. So on our peptide bond, this is what it looks like. And remember that this oxygen is a partial negative, nitrogen has a partial positive because we have an electron pair that is delocalized. So since we have these opposite charges within our alpha helix and beta plated sheet, these opposite charges can take part in hydrogen bonding. Now it's important to note that in secondary structures, the R groups do not interact. That is not one of the driving forces to get these two structures. So these two structures can then obviously be disrupted by having too many bulky groups, having electrostatic repulsion, and things like that will disrupt the structure. And then we have our tertiary structure. So within our tertiary structure, we can have alpha helices, we can have beta plated sheets, we can also have disulfide bonds. And remember that disulfide bonds are simply two cysteines that have been deprotonated at their sulfurs and they form that bond right there. So once again, tertiary and quaternary both can have beta-plated sheets, alpha helices, and these disulfide bonds. Both of these tertiary and quaternary structures are driven by the hydrophobic effect. So essentially, the hydrophobic non-polar molecules will cluster in the center, whereas the hydrophilic polar molecules will cluster on the outside and interact with the water because that is more favorable. So what's important to know, tertiary and quaternary structures, both of these structures also exhibit hydrogen bonding on their peptide backbone, but they also exhibit hydrogen bonding on their R groups. And that's what makes these two different compared to secondary. Secondary, we see no R group interactions via hydrogen bonding or electrostatic. But for tertiary and quaternary, we see hydrogen bonding at the R groups of amino acids and the backbone of the peptide. We also see electrostatic interactions between oppositely charged residues. So obviously the disruptive forces for these two structures would be electrostatic repulsion, bulky groups, the deprotonation of these cysteines to break the sulfide bond. That would also be disruptive of their structure. What's also unique to tertiary and quaternary is that the negative charges can also be stabilized by metals. For example, zinc or magnesium, they have a positive 2 charge, so they can stabilize any negatively charged oxygens or such in both of these examples. So what's the difference between tertiary and quaternary? They have so many similarities, but what makes them different? So essentially, quaternary is made up of 2 or more tertiary structures. So we get our tertiary structure, and then when we have 2 or more tertiaries coming together, we get quaternary structures. Now, you may wonder, how do 2 tertiary structures interact? Well, remember that on the outside, we actually have some hydrophilic polar amino acids. So that could be, for example, we might have glutamate on the outside over here, and on another tertiary structure, we may have lysine. Now, lysine is positively charged, and glutamate is negatively charged, and those 2 tertiary structures will be able to interact with one another due to electrostatic interactions. And when they come together, They'll form a quaternary structure. So essentially, these are the four structures that we should understand and know regarding proteins, polypeptides: primary, secondary, tertiary, and quaternary structures.